friends and my dear students. Myself Professor Mundhe Sandeep, Head and Assistant Professor Department of Microbiology, Sanjuni Mahavidyalaya Sapuli. So today we are going to see the BSc first year third semester microbial staining techniques. In this unit we will see the next part, that is the gram staining procedure. In the last period we already studied the monochrome or simple staining method. This is the second staining process or staining techniques, that we can say gram staining techniques or gram staining methods. This is the common gram staining methods. It is important because why it is called this method is also called as differential staining technique. So we can differentiate two different types of microorganism in one staining process. And first time the scientist bacteriologist Hans Christian Gram in 1884 discovered this method, and this is very important because with the help of this method we can differentiate two different types of bacteria, gram positive bacteria and gram negative bacteria, which helps in the classification and differentiation of microorganisms. In the next or in the later on we will see the what is the difference between gram positive bacteria and gram negative bacteria, what is detailed information about that we will see in the next. But before that, this is the staining techniques. So after that, what is the basic principle of this gram staining? When the bacteria is stained with the primary stain crystal violet and fixed by mordant of some bacteria are able to retain the primary stain and some are decolorized by alcohol. This is the principle where how the bacteria colorize and how the decolorized bacteria for next this further microorganism staining process. The cell wall of gram positive bacteria have a thick layer because which generally contains proteins and sugar complexes, and that we can say peptidoglycan, which also contain some less amount of lipid also, and they are very low. Decolorization the cell causes the thick cell wall to dehydrate and shrink, which closes the pores in the cell wall and prevent the retain stain from existing the cell. This is the basic principle. So the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram positive bacteria and appears blue or purple color. Now we'll see the requirement for this method or this technique. Actually, this is the practically based theory paper, and when we'll in offline classes and offline period we'll see our practically this process or these techniques, how we can carry out this process or these techniques with the help of and how it is possible. So requirement, it is a crystal violet, it is this one of the stain. Second one is the iodine, the mordant stain. Third, decolorizing agent, that is the acetone or alcohol, means ethanol 95%, and safranin, it is the counter stain. And apart from that, we can also use a glass free slide, new chrome wire loop. This is also one of the requirement. Now the procedure, how can we carry out this gram staining process? First of all, you have to take one grease free slide, means clean slide, in one hand in this way, and with the help of another hand you have to take the new chrome wire loop, and with the help of that new chrome wire loop you have to take the sample of bacteria, culture of bacteria, and keep in the center of the slide and prepare the smear by spreading that sample on grease free slide. After spreading, you have to remove the new chroma loop, and whatever the smear on the slide, you have to air dry. You just keep the slide for 1 minute in the air and heat fix. Heat fix means you have to keep the slide on the above the gas burner, above the gas burner, due to which what happens, whatever the smear on the slide, it is a heat fixed, it is dried completely. And after the drying of smear completely, you have to add first stain, that is the crystal violet. You have to just add 2 to 3 drops on the smear of the crystal violet, and for 30 seconds or 1 minute. After completion of 1 minute, you have to remove that what stain by under the slow running tap water. After that, you have to add grams iodine. This is the another stain on the smear. Keep for 1 minute, and after completion of 1 minute, you have to remove that stain again under slow running tap water. After that, drop wise, you have to add 95% ethanol on smear for decolorization process. After decolorizations, you have to add final stain, counter stain, that we can say safranin, or in place of that we can also use basic fuchsin. You have to keep that saffron or basic fuchsin for 1 minute on smear. After that, you have to remove that safranin or basic fuchsin under slow running tap water. After that, you have to air dry the smear, and after air drying, you have to add a one drop of cedar wood oil on the smear and observe the slide under the oil immersion objective microscope. Under the microscope, this is the observation process. We can observe these two colors microorganisms. Gram positive bacteria, they looks like purple blue color, and gram negative bacteria, they looks like red or pink color. It means that we can differentiate two different types of microorganism in one sample of microorganisms. It means that one is the gram positive bacteria, another one is the gram negative bacteria. That's why gram staining method or technique is also called as differential staining techniques, because we can differentiate gram positive bacteria and gram negative bacteria. So this is the method, this is the technique where we can differentiate the two kinds of microorganisms. So friend, this is all about the gram staining process. So in the next lecture, we will see the another staining techniques for observation of microorganism, because you know already about microorganism, they cannot see with our naked eyes. For observation of such microorganism under the microscope, staining process is important, because microorganisms are basically colorless. Without coloring, the microorganisms cannot observe under the microscope. So friend and dear student, you are the student of microbiology. Already I will given idea about the COVID-19 disease. You stay at home, don't go anywhere. If you are going outside the home, you have to wear mask, use sanitizer, and keep the social distance. These are the basic important things you have to use. And not only use, you have to give the information about these things to other peoples, because nowadays what happens, delta plus is the virus of corona is spreaded in all over the world, and we can't say what will happen in the future. But nowadays with the government of Maharashtra also taking the precautions about the lockdown, and that's why I'm saying you have to use these what precautions for safety purpose. Till then, bye, and we'll meet in the next lecture. Till then, bye.